And I'll say a little bit more about Beth in a second. I don't want to trot on ground that Jen has already gone through, but a few comments about Spike Coles, who was our 9th president, and himself an accomplished chemist, as is Beth, and a champion of the faculty while he was here. At the center of his mission was recruiting outstanding teacher scholars. No doubt he would be deeply proud tonight, and supported them both in their research and their teaching. And during his time at Bowdoin, the faculty here became known as one of the finest in the nation. After he left, he spent 15 years heading a company that helped to fund and bring to fruition scientific ideas and technological innovations. And in 1998, 6 years after his passing, that company, Research Corporation, and its sister Research Corporation Technologies, endowed the professorship in his name. Now, no one embodies this ideal of the teacher scholar, the one that Coles championed so well, better than Beth Stemmler. She is a cutting-edge analytical chemist. Her research is of profound importance in the field. And at the same time, she's opened the doors of her lab to Bowdoin students to an extraordinary degree. As an example, over the last 15 years, Beth has published 23 articles in leading chemistry journals. And of those 23, 19 were co-authored by her students, all of whom received recognition for their contributions in print. Another example is that when she goes to conferences to address peers, the annual meeting of the American Society for Biochemistry and Molecular Biology as an example, she often brings students to co-present with her. And again, over the last 15 years, she's done this 44 times, and in 44 of those presentations, 35 have included her students. She's also been extraordinarily active in mentoring her students. Over 100 Bowdoin students over Beth's time here have benefited from her guidance. And her research is of the highest caliber, great importance in her field, and supported by some of the most extraordinary funders in science. The National Science Foundation, the American Chemical Society, the Oak Ridge Institute of Science and Education, the Arthur Vining Davis Foundation, among others. The fact that Beth has invited so many Bowdoin students to participate in this robust research agenda, that she's been able to nurture their interest and their love for science, and continues to make such a profound impact in her field, is a testament to her unique success as a great teacher scholar. I am very pleased to congratulate and to welcome our speaker tonight, the James Stacy Coles Professor of Natural Science, Elizabeth Stemmler.
Thank you so much, Clayton, and thanks to everyone for coming. I have to tell you, I am not used to standing behind a podium. I like to walk around, so we'll see how well I can manage this. Can everybody hear me? Okay. So putting together this talk was, in all honesty, pretty challenging. I do research in a pretty technical field. I'm an analytical chemist, and actually the individual that really inspired me to go into analytical chemistry, Tom Wenzel, is here tonight, so I was really so happy to see him. I do work in an area called mass spectrometry, and so tonight I'm going to try to give you, teach you a little bit about mass spectrometry in case you aren't really very familiar with it, and share some stories about my path to where I am today, and share some of the students who've worked with me and recognize all of their efforts. So to get started, mass. So my world revolves around measuring the mass of molecules. And I just wanted to do a little calibration so we can be on the same page about what we're talking about. So I've got some molecules here spanning some different sizes, going from carbon dioxide up to a big protein. And this kind of spans, the range. We go a little further, but these are some of the challenges that analytical chemists face when they're trying to analyze matter, analyze mixtures of things. And I'm really interested in organic things. So you don't see any metals up here on the page here. My focus is really organic molecules. So mass, what is mass? Well, we step on the bathroom scale. We're measuring something a little different, and we're measuring something that is much lighter. And so one way we can look at the mass of molecules is to display their masses as kilograms. And when you look at masses as kilograms, they are very, very tiny things. But this is really what we're measuring with mass spectrometry. We are measuring the mass of individual molecules, and they don't weigh a lot. Now, in my talk, because these are such tiny numbers, we scale things up so that we're thinking about moles of material. And this gives us numbers that we can talk about. But remember, we're really thinking about measuring these very light, tiny molecules. But this is the language that we speak when we're talking about mass spectrometry and the masses of molecules. So 44 to 66,000, think about that as a range. So I've included these molecules because they also span a range of properties. And so they're going from very light to something that's very hefty. But we want to weigh them. And in a mass spectrometer, one of the basic requirements is that we have to have something with a charge on it. We have to make something +1 or -1. And so we usually start out this process, or one of the most conventional, standard ways of doing this involves taking a molecule. So here's my lovely molecule here. And we put energy into it in some way. And I just turned off the projector. Let's try that. So energy in, and our goal here is to get rid of an electron. So we ionize. And it's really that ionized, that charged particle that we want to target for our mass measurements. So we also have some other constraints on our system. The most common way to make ions, and this is the way we made them a long time ago when I was starting doing mass spectrometry, was with an electron beam. It continues to be one of the most common ways to do this. And we have this other basic requirement that things have to be in the gas phase. So we're thinking about making an ion with gas phase molecules. And just right off the bat, that is going to preclude us from being able to analyze things that are big and polar. So we've just restricted our world of things that we can target in our analyses. But this is the way things were for many, many years. Oh, and I also wanted to point out that when we're making our ion, we lose an electron. An electron weighs next to nothing, so it's not really changing our mass, and we will often just ignore that. So the other thing we have to do in order to position things so we can determine their mass, we have to get our ions into a mass spectrometer so we can operate on it with electric fields or magnetic fields, and that requires things being in a vacuum. So in my cartoon here, the gray is the air around us. And in this container, which would be the inside of a mass spectrometer, we first have to pump out all the air. And that's when our ions can exist, because if they are bumping into molecules of air, it's going to mess up our ability to measure their mass. They may change in mass, they may change directions in ways that we are not wanting them to. So vacuums are important parts of mass spectrometers. So I'm going to just show some cartoony depictions of three different types of what we call mass analyzers. So this is how we're weighing molecules once we have them ionized. And the three that I'm illustrating here are three of many types of mass analyzers. And I've picked these because these are three that I've worked with in my research, and they're three that we have here at Bowdoin. So the first one, which is the kind of lowest cost mass analyzer, it's the one that most people use. Of course, it's the most complicated to understand. So it's a mass analyzer called a quadrupole, and you can think of this as a filtering device. So a quadrupole is something where we're going to be applying some potentials here. I have vastly simplified this, but we apply time-varying potentials and some constant potentials, and with the combination of all these potentials, we can set things up so we only allow ions with a certain mass-to-charge ratio to make it through to the detector. Okay? So these are called ion stability analyzers because they make ions do these complex dances, and only ions of the correct mass-to-charge ratio get to the detector, and we dial through and scan to measure a mass spectrum. The next mass spectrometer is one that's in a magnetic field. This is my favorite mass analyzer. When we put ions into a magnetic field, as many of you know, they're going to undergo what we call cyclotron motion. And the instrument that we call, God, all of these instruments have very long names. This is a Fourier transform ion cyclotron resonance mass spectrometer, which we will call FT-ICR for short. This is an instrument that allows us to analyze ions based upon their frequencies. And I have done a very inadequate representation of what's happening, but what I'm trying to represent is where you have ions undergoing cyclotron motion, and what we actually detect is we detect the frequencies of the ions through the image current that's detected by plates inside of the instrument. And what I've also neglected to include is we have to use something called a mathematical Fourier transform to figure out what the frequencies are, and then we turn those into masses. But just trust me, this is a way that we can figure out the mass of ions. So that's our FT-ICR. And then the last instrument is really the easiest to understand. It's something that's called a time-of-flight mass analyzer. And this is a mass analyzer that determines mass based upon basically a race from the beginning to the end of a flight tube, where we give all of the ions the same kinetic energy. So we start the race, and the ions that have lighter masses get to the detector first, heavier ions last. So we measure the flight time, and we can relate that to the ion velocity and to their mass. So these are the basic ways, 3 of the basic ways that we can determine the mass of an ion in a mass spectrometer. And we measure a mass spectrum. So our output is a plot of abundance, so how many ions we're detecting on the y-axis, and what the mass-to-charge ratio is. And we may show things as peaks, or we might show them as lines. But this is what I spend my life looking at, are graphs like this. And these graphs tell us all kinds of amazing information just based upon mass. So one of the ways that we use mass spectrometry has to do with how ions behave when they get a positive charge, or if we give them some energy. Ions, again, gas phase, all by itself, in a mass spectrometer, in a vacuum. This positive ion, which may have an unpaired electron, it's like ready to do some chemistry. And in mass spectrometry, we're often doing chemistry without having things react with anything else. It all happens internally. So the molecule can undergo rearrangements, bond cleavages, and we make fragment ions. So we can break bonds and make different pieces. And we may have some of the undissociated, unfragmented ion left. And all we see are the things that have charges. Things that have no charge are invisible to us. So we get a mass spectrum. And if we're lucky, we can figure out what was the ion that started everything. So that's what we call the molecular ion. It represents the molecular mass. And then we get these pieces that are called fragment ions. And what's really fun about mass spectrometry is this now presents a puzzle, because we're trying to piece together all of these things that we can see, and our goal is to then relate that to what was the original molecular structure, okay? And for me, that's one of the most fun things about mass spectrometry, is that puzzle aspect. You're really just trying to solve puzzles all the time, but just based upon mass. Now, if we think about having a mass spectrum of one chemical, but then it's mixed with something else, life gets complicated. And think about having it mixed with 100 different things, then it's really crazy and complicated. So, and it's particularly complicated recognizing that all we're measuring is mass. So we typically try to make our life more sane by physically separating things before we do the mass spectrometry. So mass spectrometers are almost always coupled with chromatography, where we separate components of a mixture. So we're physically separating things, and we get a different kind of output, which is we're looking at a function as a function of time at what's coming out of the column. And this is normally how we're introducing samples to our mass spectrometer. There's different compounds. We can do it with what's called gas chromatography or liquid chromatography, GC or LC. There's going to be a test at the end of this lecture to see who can tell me the acronyms. Maybe you could just count acronyms. We have an amazing number of acronyms in my field. So we have GC and LC, and again, we're thinking about what range of molecules can we actually access, and with GC, we're also not able to analyze anything that can't vaporize, so no proteins, no sugars. Liquid chromatography is much more compatible with a wide range of things. So we have these two options for separation. But what we get is a two-dimensional data set. So we're looking at data where we have our separation as a function of time, and we've got the mass to charge ratio and abundance. Okay, so it's multidimensional. So to just introduce what you can do with mass spectrometry, I'm going to describe something that's a project that or a lab that we do in one of my classes. It was derived from a student project. And this is where every year we gather $20 bills. Sometimes they come from faculty, sometimes students bring them. But the goal of the lab is to extract whatever is on those $20 bills, and believe me, there's a lot of stuff you don't want to know about on a $20 bill, but we do this to determine cocaine. So it turns out that currency collects alkaloid drugs, and so these bills can be sort of, indicator dollars of what alkaloids may be in use. So to do this lab, we extract all the organic compounds from the bills, and we do some cleanup that I'm not going to tell you about. And then we analyze things with gas chromatography mass spectrometry. So we vaporize things and we figure out the mass to charge ratio, measure mass spectra. But in order to do this, we really need to know the behavior of cocaine. So the start of this experiment requires purchasing or acquiring a pure standard of the analyte that we want to analyze. So every year, and Clayton, you may remember this from last year, I came to your office and I said, President Rose, my students and I really need some cocaine. Will you buy us some? And Clayton, he's like, Beth, whatever you want for your students, I am so happy to provide this. So Clayton is shaking his head like he doesn't remember this conversation. But we very legally, by DEA exempt standards of the drugs that we're trying to analyze, and having that standard tells us, gives us information about what we should be looking for. So it tells us from the separation as a function of time, it tells us the time, and we can look in our multidimensional data and we can get a mass spectrum, and it tells us, what is the personality of cocaine in terms of the fragment ions that it produces. So we have that information, which is acting like a fingerprint, so that tells us what to look for. And then we can go and we can analyze our actual drugs. We also like this is the fun part. This is the kind of organic chemistry, thinking about how ions fall apart. This is what cocaine does. But this is what a bill extract looks like. So many peaks, a lot of things going on. We use our standard, and that tells us where we should look as a function of time, and then we can look for evidence of cocaine through the mass spectrum. So that's our way of confirming identity. Okay? So this is an example of how mass spectrometry is used for analyzing things. And when we do this lab, we also do it very quantitatively. We're figuring out how much cocaine is on dollar bills, and we can get a number for that. So how does that connect with my research? So that's kind of setting the stage. My research is directed at trying to find ways to either figure out how we can make mass spectrometry more selective. What are things that we can do which may be related to how we ionize molecules, it may be related to how we get them to fall apart into pieces, but what are things that we can do so that we can look at a very complex mixture and say, oh, that's what I'm looking for, and ideally without the need to have a pure standard that we have to buy. Ideally, we would be able to figure out how to get that really specific information just from knowing things about maybe an attribute of the molecule, a particular part of the structure. And we like looking at patterns. So are there ways that we can use patterns of information to learn more about molecules? So I'm going to just start to introduce this idea going way back to when I was in graduate school at Indiana University, where I got to briefly play with fish. So my work was very directed at fundamental aspects of mass spectrometry, but my research advisor said I could not graduate unless I analyzed something real, and so I had to work on a project where we analyzed fish. And we were looking for chlorinated pesticides that were present in fish. These fish, if you're from Michigan, these are fish from rivers and tributaries that led into Lake Huron. And we were trying to use carp as indicator organisms. So they're they stay put, they don't eat other fish, and so they should be sort of in chemical equilibrium with the environment. So I got to grind up fish. And when I tell my students about this, I love to bring up the basematic. How many of you are old enough to know about the basematic? So grind fish and do a bunch of things to purify it. And this is what you see if you look at a fish, just make a positive ion the way I described and analyze it by GC mass spec. You make the ions after you do the GC. And so we see a bunch of things, a bunch of peaks. There's a big peak for squalene. There's a peak for alpha tocopherol, vitamin E. But none of this were the things we were looking for. We had an overwhelming amount of other components that were present. And so this is where we need selectivity. We want something that will allow us to see the things we want and not see all the stuff we're not interested in. So to be able to see our contaminants, we worked with negative ions. And this was my introduction to the world of negative ions, which I embraced for many years of my research career. This is a more selective way to make ions. So instead of making a positive ion, molecules that have positive electron affinities have the ability to hold on to an electron, can capture electrons, and we make a negative ion. And this is a capability that not all molecules possess. So a lot of things like squalene are not going to be able to form a negative ion. So this is what we saw for a fish. Very different chromatogram. We were seeing individual peaks, and these are all chlorinated components. And we could see, for example, DDE. So this is an example of something that's a selective technique. And so I did, in my graduate work, a lot of investigations looking at how do negative ions fragment. So I've always been sort of a wannabe organic chemist. I really like thinking about molecules and how they fall apart or interact. And so I got to do that in my graduate work, and I went in postdoc and was looking at other ways that we could be selective about ionization. And so negative ions. And I came to Bowdoin. I was, applying for jobs, and when I came to Bowdoin for my interview, it was pretty intimidating in terms of what it would be possible for me to do at Bowdoin. My work really involved a lot of fancy instrumentation that wasn't available at Bowdoin. And I'll never forget, so Ron Christensen picked me up at the airport for my interview. And he, in our conversation in the car, basically said, be ambitious, go for it. This is not something that's like typical at undergraduate institutions, but it's possible. And that was the message that I really got from all of the faculty. It was really encouraging. And so I started at Bowdoin, and we got a mass spectrometer. And this was a research grade mass spectrometer. We could make positive ions, we could make negative ions. It had the ability for us to add gases and sort of play games with making ions. This is a picture of my mass spectrometer. And those of you who were around in the chemistry department at that time may be looking at this picture and just having this, like we may be having a mind meld. Okay? And it's not a mind meld about the student. Let's see how many people are remembering. Oops. Okay, my pointer's not working. Oh, there. But that carpeting. Who was thinking about the carpeting from the old days in Cleveland Hall? This is before Druckenmiller. The chemistry building was old. Students did microscale organic chemistry in part because there were no fume hoods, no student fume hoods. And the lab where I was putting my brand new special instrument was this lab. Most labs have like chemically resistant floor tile. This was a lab that had been lovingly carpeted with carpet acquired by our custodian, Joe Carlo. And he had found this just awesome tiger striped carpeting. So how many people get to move into a lab with tiger stripe carpeting on the floor? One of the first things we had to do was cut the carpeting out so my instrument with its 4 pumps didn't drop oil all over the floor. But this was my first instrument. And what I spent a lot of time doing, and what my students and I spent a lot of time doing, were thinking about the kind of location where we were making ions. And this is our ion source, where we would add gases and we would ionize here. And it was basically like a steady state reaction chamber. And so some of the work that we did had to do with redesigning, optimizing our ion source. And Bob Stevens and Searles made us amazing little tiny ion sources that we used for our work. And we modeled things, and so a bunch of things trying to understand the dynamics of what was happening in the sources and how we could control the chemistry. And we spent a lot of time looking at gas phase ion molecule reactions. So now, encouraging molecules to react, encouraging an ion to react with another molecule instead of this falling apart in isolation. And we looked at things that are some of the things, the ions that are conventional, things you might encounter in a chemistry lab in solution, but other things that we really could only access in the gas phase. So we looked at things including hydroxide oxide with 1 unpaired electron, superoxide, and we were thinking about how things reacted and mechanisms. So this is, again, trying to be an organic chemist. We could use isotopic labels to sort out reaction pathways. We made some really cool ions. You're getting excited by looking at this right here. This is a really cool ion. This is really cool because it's got an unpaired electron right there and a negative charge over there. It's got a great name. It's a dystonic ion. Okay. Wow. So we made that. And we looked at how we could use these things analytically. So just really quickly, for just one example, we were looking at thinking about phthalates, which are compounds that are pretty ubiquitous in a lot of materials and are of environmental concern. They are found in a lot of plastics and containers and medical devices, and they're not covalently attached, so they leach out of those materials. And when you analyze those by making positive ions, this is kind of a head-to-head of two different molecules. When we make positive ions, do those mass spectra allow you to tell the two compounds apart? They have just very subtle differences. So this was not as helpful as getting something that was more specific to the molecules. And so this is where, in reactions with superoxide, we were able to distinguish them. And we were also just really interested in superoxide. How does it react? And I'm not going to include any information here, but we were able to find evidence to support this being a really good SN2 nucleophile, so playing like an organic chemist again. But where I really got to play like an organic chemist was when I would travel back to Oak Ridge National Lab, which I did a lot when I first started at Bowdoin, because they had this really amazing type of mass spectrometer. And it's a mass spectrometer where you can act like a synthetic organic chemist, but you don't have to go into lab and mix together chemicals. You can just sit in front of your instrument and conduct your work. So let me explain how that happens. So this is the type of mass spectrometer where you have a magnetic field and the ions are undergoing cyclotron motion in the magnetic field. So the first thing we can do is our synthesis. We can make some starting material. And we do that with ionization. And then we decide, it wasn't a pure synthesis, I've got a mixture of things, so I want to do a purification, which usually requires a lot of work and physical separation, but we just send in our cyclotron frequencies for the ions we don't want to see, and we eject them. They're gone. Takes about a microsecond. Then we can think about, oh, I want to do some chemistry, so I can do a chemical reaction by inletting a neutral gas, and I can let reactions take place. I've made a molecule that looks like a fly, but we can make products, and then we can analyze those products and figure out what we made. We have to get rid of the gas. We have to do it at low pressure. We excite the ions into coherent motion, measure their frequencies, and then we can decide, I'm done with my experiment, but instead of washing any glassware, we just eject the ions. So we eject them and they're gone. So chemical synthesis in an instrument in an Fourier transform ion cyclotron resonance mass spectrometer. So this is a really cool instrument, and we use this to validate some of our data where we could look at kinetics for reactions, where we could pick out superoxide all by itself and look at its reactions. But we didn't have one of these at Bowdoin. And at the same time, there were crazy things happening in the field of mass spectrometry. Because remember how I said we couldn't look at proteins, we couldn't look at really polar molecules? Well, two ionization techniques were developed, and they really came into active use and publications right when I was starting at Bowdoin. And these two techniques, for some more acronyms, are MALDI and ESI. And they allowed mass spectrometrists to measure a much wider range of compounds. You could look at very large proteins. You could look at very polar molecules. So MALDI, which is matrix-assisted laser desorption ionization, kind of a mouthful, is a laser-based ionization technique where you pulse a laser and you have a solid material. You have your analyte embedded in what we call matrix. And that really rapid heating beats out thermal decomposition that would otherwise prevent us from being able to measure the M/Z of a protein. The other technique is electrospray ionization, where we start with things in liquid form and we put them in a high field. We make a spray, and in this spray with drying gas, we form gas phase ions. So these are two techniques that all of a sudden were available for ways to look at a wider range of molecules. And we didn't have any of these at Bowdoin either. And I, during my junior faculty leave, went back to Oak Ridge National Lab, where I worked with Michelle Buchanan, who is my postdoctoral advisor, and she was really a visionary. She was an early adopter of ion cyclotron resonance instruments, which weren't very popular, let's say not very popular, but they were really intimidating instruments. You had to know a lot about Fourier transforms and frequencies. And when I was working on the FT-ICR instrument at Oak Ridge, you know, there were less than 100 of those instruments out there. And I worked with two scientists, Greg Hurst, who was a spectroscopist, a physical chemist, and Bob Heddick, who did mass spectrometry, and got to build a MALDI time of flight mass spectrometer, and got to do some of the early work trying to make MALDI work with FT-ICRs. And it was super fun. I have to, if any of my students look at this top spectrum, which is a MALDI time of flight mass spectrum of some DNA adducts, a DNA adduct. This is resolution with peaks here that is horrendous. This is like the ugliest mass spectrum, but I measured it. And this was kind of the state of the art for older instruments at the time. And in contrast, the instrument on the bottom, which I've now changed acronyms, this is the FT-ICR, which we sometimes call an FTMS. This is very high resolution, the peaks are very narrow. And with this instrument, I had just great fun. I was going into lab every day, just chomping at the bit. It was so fun. It was so great to be able to, you know, you had so many experiments you wanted to do. It was a really productive time. We were able to sequence pieces of DNA that were up to 12 bases long and identify covalent modifications on different bases. So it was just really exciting. Lots of chemistry. And it was great to go back to Bowdoin to be with my students and colleagues, but you know, it was sad to not be back at Oak Ridge. And at the same time, my life was also changing in a really wonderful way because we started a family. I think when this paper came out, it was the year my son Daniel was born. And I just couldn't really go travel to, you know, bop off for a while to do experiments. And so we were continuing doing research at Bowdoin, and Ron Christensen walked into my office one day and said, you know, NSF, the MRI program, is dropping its matching fund requirements for undergraduate institutions. You should write a proposal. I was like, I'll ask for an FT-ICR. No undergraduate institutions had one of these instruments. This is really like a big step. And amazingly, they funded us. So we, in 2002, had our own FT-ICR instrument. It was like I had my own, you know, candy shop next door. And this instrument has really been an avenue for me to collaborate with a lot of people at Bowdoin and in other areas, coming from other areas. It's like an incredibly cool instrument. I'm going to just tell you a couple of stories about how we've used this. Just to do, this is like talking about your new car. Have to, you know, show off the attributes of the instrument. We have a 337 nanometer pulsed nitrogen laser that does our desorption. Our ions are transmitted to the, whoops, oh dear, we went way, am I going in the wrong direction too? Woo! How did that all happen so fast? No, I have no idea how I did that. I'm going the wrong way. Okay, I'm totally going the wrong way. Sorry. Yeah. We would, so I'm going to just describe it while I'm talking here. So, clicking here. I think I've had too much caffeine. Ion guide. So we would make our ions. We have a 2-meter ion guide that transmits the ions into our magnet, and there we trap our ions. They undergo cyclotron motion, and we can do all of that stuff, that organic chemistry. We can play games with the ions once they're trapped. So this is one other thing that's important about this instrument. This instrument is so cool, it's hard to just capture all of the neat things about it, but one is that it has the highest mass resolution of all types of mass analyzers. So resolution, we're thinking about measuring mass, and when we have low resolution, we can't separate mass very well, but when we have high resolution, we get really good distinction between peaks. And this is really useful in chemistry because when we can know the mass more precisely, we can know more about what elements are making up our sample. So we get more digits for measuring the elements, and when we put the elements together to make molecules, this is really useful. So if we have something that has a mass of 44, for example, and we're thinking about how much carbon, hydrogen, and oxygen weigh, we have a lot of possibilities for things that can be 44, different combinations of those elements. And while you might not really think about hydrogen as being a heavy element, I think about hydrogen as being heavy because hydrogen, in terms of what digits are coming after the decimal point, it is bigger than 1. And when we think about oxygen, oxygen is less than 16. So oxygen's light, hydrogen's heavy, and we can distinguish things based upon those differences in the elements. So we get a lot of information when we can do high resolution. This is an example. This is actually Daniel's breath, by the way, when he was in elementary school. Daniel's breath measured at low resolution, really fat peaks on our quadrupole mass spectrometer. Daniel's breath at high resolution, measured on our FT-ICR instrument, where we can zoom in and zoom in. These are things that are both 44. And we can distinguish things. So that's high resolution gives you power. Just a couple, I'm not going to go through a lot of the different things that we've done on this instrument, but I just wanted to point out sort of 2 things. One, this is some work coming from a collaboration with Ron Christensen, and one thing that I really found very fun is this was some work where some graduate students who are working with Richard Schrock at MIT came up to Bowdoin to work with my student, Lucas Amundsen, to do some characterization of some polymers that they were making. So it's not really, it's sort of a flip thing. How often do MIT students come to Bowdoin to use instrumentation? And so we were looking at things that were bigger, so what you can kind of take home from this is everything we look at, they're mixtures. I haven't really mentioned chromatography because we're not doing chromatography. This is a technique where we look at everything together. And where we really put this into action, into some really fruitful work, was in some collaborations that have been ongoing collaborations with Patsy Dickinson from Bowdoin and Andy Christie, who's at the University of Hawaii, who is remarkably here tonight. But he didn't really travel here just for my talk. But Patsy and her students have expertise in doing electrophysiology. They can dissect out the nervous system of crustaceans, which are really great model organisms for understanding pattern generators and how rhythmic motions, movements happen in our body, and are interested in how signaling molecules like peptides, neuropeptides, can modulate that behavior. So make you walk faster or slower, for example. And Andy has expertise with imaging and with bioinformatics and genetics. And so we put our resources together where with my instrument, we could look at neuropeptides. And we could, through identifying neuropeptides, give Patsy and her students molecules that they could then test to see what's their activity in the nervous system. And if Andy wanted to be looking from an imaging perspective, knowing what the peptide was is another important element of doing any kind of imaging where you have to generate antibodies to do the imaging. And even when we had genetic information, when Andy would look at genetically and say, there should be this peptide, peptides undergo post-translational modifications. So this is a peptide. I've now switched to letters which represent amino acids. And the peptides can change their structure in ways that are important for their activity. So here's what we would do. We could take a very teeny piece of tissue from the nervous system of a crustacean. So see those tweezers, see those fingers? This is like not a lot of material. We put this on a probe tip with matrix. We crystallize it, and then we stick this into our instrument for analysis. This is Maureen Guiney, who beat her head against the wall for most of a summer trying to get this to work as we were first developing these techniques, and then she was successful. And we would fire a laser at our sample. We could store up ions in our cell, which was a great way to get better sensitivity. And we could look at individual signals from individual tissues from individual animals in ways that were really sensitive, that were kind of a departure from a lot of work that had come earlier and was sort of a harbinger for things that were coming down the line in terms of sensitivity. And I have sort of two little stories here. And one is something that was never published, but was kind of a fun story. This is the saga, the story of the Piphamid. So this is a hermit crab that, if I get the story correct, Andy collected from near his parents' house in Connecticut, which was right near a nuclear power plant. And so we see a bunch of neuropeptides, and one of them is this glyciphamid. So this is something we see in a lot of crustaceans. This was animal 1. Animal 2 had a partner there, and the fractional mass, this is kind of the fingerprinting idea, we knew that this was related to G-ciphamid. And we could, with the instrument, measure the mass. So we've got really good ability to measure mass. And from that mass, we could say, oh, that looks like a serine to proline amino acid change. And we could break this peptide apart, which is a strategy that we use to figure out amino acid sequences. And by differences in the mass, we could tell what parts of the peptide sequence were the same and what were different. And we knew it was a piphamid instead of a ciphamid, where the P had replaced an S. So this was an example of something where we've seen in some work where looking at individual animals, you see differences, which can be an issue because people often pool together tissues. So if we want to really learn about the behavior or the complement of peptides from one individual, we need the sensitivity to be doing it one individual at a time. And the other little story here is just about patterns. And this is a story about a family of neuropeptides called orcokinins, which give very predictable patterns of behavior, where we can look at masses, look at the mass differences, and use that to say, oh, this is an orcokinin. And we can sequence to get the component of the orcokinin that varies in sequence, where we've used the mass because some of it stays the same. And Chris Cashman, a student that worked for us, looked at a whole huge array of organisms, and by this kind of pattern recognition approach was able to pull out what orcokinins were present. So this is just basically a big table that says Chris found a lot of orcokinins by pattern recognition. Okay. So this instrument was an amazing instrument. It's been a part of a lot of work that was done at Bowdoin and that's been done collaboratively. But we were also recognizing that there were some limitations associated with it. And we had new faculty coming to Bowdoin who really needed to be able to use mass spectrometry, but needed to separate things before they did their analysis. And so we were able to justify our latest edition. So this is kind of the story of 3 instruments, and this is our most current mass spectrometer, which is something that now uses liquid chromatography. So we went from gas to no chromatography, and now we have LCMS capabilities. So this instrument is an instrument that is composed of, so we've got a liquid chromatograph, an ion source, a mass analyzer, a detector. So this gives us access to a really wide range of molecules, And it also gives us access to so much information. We've really entered this like informatics, bioinformatics world. Just to tell you a little bit about the instrument, we use columns to separate our mixtures. And when we think about, this is liquid chromatography, things in solution, liquid chromatographic columns are usually pretty wide. And we're working with columns that are just a fraction of the size of a normal column. So they're really narrow, and they have very low flows. And this translates into not needing a lot of sample for analysis. And the columns are actually constructed on chips. So these are all microfabricated. So we have these little chips, and we insert the chips into the instrument, and this is how we conduct our separations and our ionization. So there's a little tip at the end. So, and here we're doing this other kind of ionization, electrospray ionization, where we're making ions by spraying them in an electric field. Okay. So, and the instrument allows us to analyze the ions where we're using time of flight, this flight time idea, which is now in a whole different world. It's high resolution because we use something called a reflectron that gives ions a chance to kind of catch up if they have different kinetic energies. And we can break up ions to get more information. Okay. And we can analyze the products. So basically we have access to not only dimensions of information, we have 4 dimensions of information now. So we have incredible amounts of data that we can access. And we use this for many different things. We can use this to analyze proteins, where we can break proteins up by digesting them with enzymes, and we can separate them. We can see what the mass of individual pieces of a protein are, and we can sequence or get information about pieces of those peptides, and then we can rely on computers that help us do the data analysis. So this is what people do all over the world to identify proteins, and we now have the capability to do this at Bowdoin. And we can look at intact proteins. So this is the separation of really big molecules. But what I want to tell you about is actually a student project. So we've done a lot of different things with this instrument, but this was probably one of the craziest samples that we've looked at. We had students Meg and Lloyd, who decided that they wanted to see what was going into and out of the Brunswick waste treatment plant. And I have spared you pictures of the samples, but you can probably picture their color. So these were some really complex, nasty samples, and they did a lot of work to filter and to purify the samples so that we were looking at just a segment of the total mixture. And their idea was they wanted to do what's called sewage epidemiology. Has anybody heard of sewage epidemiology? It's this idea that when you do surveys to find out about drug use, everybody isn't willing to fess up. But if you look at what's being flushed down their toilets, you're going to have a direct measurement of what their drug consumption is. And so they wanted to look for illicit drugs to see if they could detect them. And we were doing a purification that was targeting alkaloids. This was the influent to the wastewater treatment plant after extensive purification. And I just thought I'd share with you a couple things that we found. So one thing that we found was nicotine. And to convince ourselves that it was nicotine, this is where we measured the mass to see if that was consistent with the elemental composition of nicotine using the high resolution capabilities of our instrument. and it matched. And we did MSMS, where we get this very specific fingerprint. So this is the fourth dimension of information. So sadly, Megan Lloyd did not detect any illicit drugs. We found a little bit of evidence for cocaine, but it was not very resounding. But we did find a bunch of other things, which may be a reflection of our population or what kind of drugs are used, but a lot of beta blockers, cough suppressant, and Benadryl, amongst a lot of other things. So just an example of the kind of data that we can acquire. And we did these measurements with a control where we spiked in 20 nanograms of cocaine in a blank to see if we could detect that. And we could detect that, taking that through our entire procedure. So the last thing I'm going to tell you about is just this idea of informatics and just the challenges that analytical chemists are facing. And one problem is thinking about, related to wastewater, is the problem of how do you degrade these persistent compounds that are not being removed by our wastewater treatment plants. And I've been doing some work in collaboration with faculty who are trying to photocatalyze that degradation process and are synthesizing new catalysts. And one of the challenges of thinking about degrading things like, this is birth control estrogen, which is a problem in the environment. Here's birth control estrogen, one with no photolysis time, and then this is after 60 minutes of photolysis in the presence of a catalyst. And there's a huge number of products that are formed. And so if we're photolyzing to decompose these pollutants, then you need to know what you're making and whether what you're making is going to be perhaps even more of a problem when it's introduced into the environment. So the challenge is these are a huge number of compounds, and so how do you access that information? And a lot of things that are very similar in their formulas. And in a similar way, we've been doing some work with biofuels that are derived from wood being synthesized at UMaine Orono. It's, again, an incredibly complicated mixture of things. So much information, so many compounds. How do you meaningfully identify things, where our old approach would be to go through one at a time, but we have thousands of things. And so this is where you need to be using informatics approaches, and we're working on using our new instrument to be able to provide some extra information where we can both take chromatographic information and MSMS information where we break things up to be able to look at what we're forming. And what we're trying to do is be able to take this huge body of data and display it graphically in ways that are more accessible. So we're thinking about, and have actually been working on doing our LC-MES, where we can get our high resolution MES measurements. We can figure out what are the elemental compositions of all the things we're detecting. We can give them a chemical formula. But we still have a problem because we have many things that have similar chemical, or have the same chemical formula, but have different properties. And I've just listed 4 molecules that all have exactly the same mass, all the same chemical formula, but they have different substituents. So the kinds of bonds they have are different. And they can progress from things that have less oxidation to more oxidation. And it would just be really nice to be able to at least say that attributed to the compound with this formula, we know something about its functional groups. And so what we're doing is we're doing chemistry to try to tag the different functional groups. So we've been targeting hydroxyl groups, where we can use isotopic tags to see differences, and we can then react our in this case, a bio-oil with these 2 different tags, mix things together, and then in one analysis, we can get information about all the compounds that are present. We can generate compound lists, we can generate formula lists, and then this is where the informatics piece comes, where we need to put all the information together and sort things out. So thank you so much for coming tonight. I'm so grateful for everybody taking the time to be here. I want to end by thanking the students that have been part of this journey. In my journey through science, I feel so incredibly lucky both to be here at Bowdoin, where I have been able to access all of this really amazing instrumentation, been able to use it to answer a lot of questions. And I have, my scientific trajectory and my experiences have been so enormously enriched because I started as a very focused gas phase, negative ion chemist looking at small molecules. And through my collaborations, I've just learned so much. It has never been dull. It has never been uninteresting. I go every day and I would just love to be playing in lab. And I've had such wonderful students that I've worked with and taught. It's been such an amazing, enriching experience. And to start, these are students that I've worked with on my initial instrument. And just want to acknowledge, so Colin Segovis was the student who found the dystonic ion. John Diener and Jen Swift, whose picture I don't have, did work with superoxide. And a lot of these students also did a lot of synthetic organic chemistry, making isotopically labeled molecules that we use to sort out mechanisms. This is phase 2, students that worked on the ICR instrument. And here's Maureen. And just want to recognize Heather Provencher and Noah Gardner, who both really moved us forward with some of the work we did in collaboration with Patsy and Andy. So they put a lot of time into developing methods to be looking at crustacean tissue samples, and there are many other students here who are involved in that. I want to highlight Chris Cashman, who did the work with the orcokinins and the pifamid. And then the most recent crew, so a bunch of students who've been working on bio-oil samples that include Matt Rasmussen, Sally Kim, Hunji Lee, Sean Morin, and I think that's about all the ones that were doing biofuels. So a lot of students have collaborated with a lot of wonderful people. Brian Nelson from Bates has been doing work with us on bioinformatics. Of course, Andy. Students worked with a bunch of grad students from UMaine Orono, and Jinsey Joseph in particular was really important for a lot of our biofuel work. And Paige Case was the one who synthesized a lot of the biofuels, or pyrolyzed wood to make the biofuels. And then at Bowdoin, Patsy, John Lichter was a collaborator who dragged me out in the field, maybe not kicking and screaming, but taught me to pay true respect to dirt and call it soil. And Anne McBride, who has taught me an enormous amount about proteins. I came to Bowdoin and got to work with a bunch of really amazing chemists, and these are chemists retired from the department. And Pam Labby, our department coordinator of 40 years. Did I get that right? Just, I don't know what we would have done without her. Dana Mayo, just an inspiring member of our department, and was someone who really just gave me a lot of energy and a lot of advice. And Sam Butcher, I don't think anybody in the department has his glass blowing skills, but an amazing instructor who was a fabulous mentor to me. And David Page and Judy Foster, and of course Ron Christensen. And I feel really so lucky to be in such a wonderful department that has transformed over the years, but I really feel like we're kind of a chemistry family, and it's a pretty special place to be. So I want to thank all of them, all the funding organizations, Research Corporation, and thank you so much for your attention tonight, and I'd be happy to answer any questions. interesting, ambiguous. So certainly, moving into this world of biology and proteins, I've been collaborating with a colleague in the department who looks at glycoproteins, and they are probably the most, in terms of being ambiguous, they're kind of ambiguous because it's not something that is a reliable chemical structural change, and I think they're like among the biggest challenges. As far as compounds that are just kind of cool and interesting, I've been really intrigued with the biofuels that we've been working on because there's really cool chemistry that happens that's kind of polymerization chemistry, which I've really enjoyed thinking about and trying to both, trying to predict it and then see if we can validate it through samples that we look at.
Awesome talk, Beth. Do you have a wish list for your next big toy? Are you going to drive the same classic convertible forever, or are you going to find something cooler?
I feel like I'm sitting in a pretty sweet seat right now. But there are always really cool things that are out there. I am so amazed with what we have, and we have so many things that we can do with the equipment we have right now. But there are really neat things that are happening in the field of mass spectrometry. And one of probably the neatest thing, the thing that gets me most excited about, are measurements that are now being made using ion mobility. So this is a little different, where we make ions, but we now have them flow through a gas in a drift tube. And this gives you ways to look at conformations of ions. So what's their kind of secondary structure? How, are they spread out or are they compact, even if they have the same mass to charge ratio? So it gives us like another, element of how you can separate things. And there's amazing, amazing work that's being done with those instruments. I may just have to go visit one. Yes.
So could you talk a little bit about how do you decide what matrix to use with different instruments with different test things?
Yeah, so one of the, the challenges with MALDI was it was really a little bit like black magic. So people have systematically looked at a lot of organic compounds. They must absorb the light, your laser frequency. So you have to have absorption happening. But there's other special stuff that happens. When we do the laser desorption, you actually have, and people have profiled these signals spectroscopically, but you're like generating a supersonic jet. So there's an element of cooling that happens, and formation of carbon dioxide is one thing that is part of the good magic. But there are matrices that people know work pretty well, and you can kind of dial in different kinds of ionization efficiencies, energies that are being deposited if you want to enhance or dial back on fragmentation. But in all honesty, it's hard to predict and say, look at an organic structure and say, oh, that's going to be a really great MALDI matrix. It almost never happens. So, did that answer your question, Sue? Yeah, and they're like the small organic molecules. They usually are things that have stickiness. They have hydrogen bonding kinds of interactions. They usually form beautiful crystals. And that crystallization and the incorporation of your analyte, your protein into the crystal structure is a key element of making that work. So you're really matrix isolating the things that you're trying to analyze.
Thank you. That was marvelous. I'm just amazed by everything you do. And one of my first experiences of that was in our wonderful collaboration, and the degree of detailed analysis you did of the mass spectra looking at the protein that we were looking at. And you just mentioned how with the new instruments, there's even
Yeah, no, it's a huge issue, and where there are good models for how things are going to behave, so in thinking about metabolism, where you might have knowledge of enzymes and transformations, or in the, the case of just analyzing proteins and protein modifications, where we have pretty robust ways of predicting behavior. The challenge is predicting it, like having some tools that allow you to extrapolate from your raw data to structures. And so the things that we are thinking about with the biofuels, I think it's unrealistic to be saying, we're gonna get structures out of this. That is the heavy duty, detailed analysis. But there are ways that we can get patterns, and I think that's really accessible. But to do it, it's a lot of data, and you need people who can handle the computing component of it. So you can have the ideas, and you can have, like, this is the flow path that I want to follow, but then it's really collaborating with people who have the computing abilities to actually make, your ideas go to fruition. I think everybody looks like they're ready to go home. Thank you so much for coming. Have a good rest of your night.